we'll start the last session here, and this was a session that we were thinking about for quite some time, and how to really put this together. And I think, given all of the technologies that we've seen just this week, but all the other technologies that we all know about, we all use, we've used in the past, we use in the present, as we also use in the future, it comes about that one of the central figures in all of this has been Lee Hood, and we see this as somewhat of a perspective talk for Lee to give his perspectives on emerging technologies, how emerging technologies came about, and then also, how does one actually have a vision for technologies and see how they can be used. And so, together with my panel members here, Alan Aderem, who's known Lee for quite some time, and also Mark Chee, who's also known Lee for quite some time, and both Alan and Lee as well. Alan and Mark have not only used technologies but also developed technologies as well, and made some critical insights as we go forward. And so each of us are just going to give a little bit of a perspective from our points of view of emerging technologies. We'll ramble on a little bit, but and then Lee will give a talk about emerging technologies from his perspective and how things go through. So I think from that point of view, I'll start off and just tell you how I got started in a lot of this. And so when I first started in 1983, I was using Edmund degradation from one of the first instruments that Lee had developed. And Lee has been a powerhouse of technology development, not only from the gas phase protein sequencer, but also the DNA sequencer, which was used for the human genome, but also the DNA synthesizer and also the peptide synthesizer. And in my career, I've used all of those, and it struck me very early on that these are technologies that are not only all-encompassing, but they have really moved the field forward. Even though back then, as a young researcher, I didn't really realize how technologies could move that fast. When we're using the protein sequencer, it was 1 amino acid an hour, and so this was almost like it was your top shelf capability of doing things. But these days, when you see these types of technologies delivering data very quickly, it comes about that technologies move on very, very fast, and you've got to have that vision going forward. I came from a place that it was very English, very stoic, and the developments of technologies was not very encouraged. And so when I had a chance to meet Lee many years ago, I always thought that would be a great place to try and interact with Lee. And so when coming here, I always thought that at least I would be at a place where some vision and technology would be able to be diffused to myself, and at some stage it might also go backwards in osmosis to work out whether or not Lee would be ever interested in any of the technologies that we were always thinking about. I think trying to understand when technology is actually needed, and also when does one actually jump into technology, takes a great lot of vision. And that's why I've always applauded Lee for his vision in that respect. And it also makes you think sometimes, is when is a good point to jump into technology, and getting that perspective from Lee is very important. And when is it to divorce yourself from technology? And one should never be totally wedded to technology all the time, because otherwise the world will pass you by. And so Lee is fairly unique in that ability to not only expand on technologies that are I suppose present in its current day, but also has a great vision as he sees a lot of technologies way down the road, much further than what many other people can see. And it takes you some time as a mere mortal sometimes to actually grasp that vision that he has. And so it's really unique to see that. And so I think this session that we will have will impart some of Lee's vision onto you, and hopefully you'll get as much out of it as I have over these years, and we hope to see that many more years to come, because Lee's not only been visionary, but he's also very tenacious, and he's also very persuasive, and it really makes it fun to work in that respect. So that's my preamble for the start. So Alan, do you have anything? Or Mark?
Yeah, so Friday, Rob asked me if I'd share some of my thoughts about Lee and technology, particularly in the context of what Lee's achieved. And I said, how much time do I have? And he said 3 minutes or 4 minutes. I said, that's impossible. I'm prepared to take on jobs, but that's not one I can lightly do, because to talk really about Lee's many contributions to technology would take us a lot longer than that. But that said, I'll try my very best to discuss the interactions that we have had in the past and the technologies that have been developed and so forth. I was at Rockefeller University for 18 years and had been thinking about the Genome Project at that time. And as Rob said, that the genome was sequenced on Lee's machines. Lee himself sequenced chromosome 14 and 15, I think it was. But as a young professor at Rockefeller, I was trying to figure out what the hell we were going to do with this, because everybody knew it was a good idea, the ones that weren't threatened about funding. But at the same time, we hadn't thought really through, I think, a lot of how we would use the information. And I ran into Lee at that time in 1992. He'd just gotten to Seattle, and I raised some of these issues, and we had a really exhilarating evening talking about this. I remember it well, despite the bottle of whiskey that sort of was drunk at that time, the two of us. But and that's something about Lee that he's really great to share a bottle of whiskey with, because he gets more and more excited, if that's even possible in terms of science. But just that single conversation actually persuaded me to leave a tenure position at Rockefeller to come out here and join with Lee and perhaps start what was then the Systems Biology Institute, which was going to happen at the University of Washington. And so a eminent scientist who I won't mention his name what the hell, Jim Darnell. He was making a counter offer to try to keep me there, and he said, you know, Lee Hood talks a lot, and he's been, 20 years ago, he said to me at the time, Lee had said that you could take a 2D gel, pick out with a robot each spot, sequence it, then essentially synthesize the genes and transform cells, and you'll be able to figure out how these things all work together. He said, you know, it's only now that he's being able to do that. And I thought to myself, Jesus, you know, what he's really saying is that this guy had the vision of what was going to happen in 20 years' time, okay? And he was seeing this to some extent as a weakness. So leaving Rockefeller was the easiest thing I ever did. And I came out here to join Lee. And the other reason I basically decided to do that was I ran into quite a prominent immunologist, Max Cooper. And I asked him about, you know, what do you think, Max, should I do this? And he said, well, nobody has ever gotten rich betting against Lee Hood. So I decided immediately I'd come out here, and so I did. We co-founded the Institute of Systems Biology, I guess it was 2001. That was Lee, Rudy Eversalt, and I. And one of the mantras that we had right from the beginning was that biology drives technology and technology enables biology. And the idea here was that you start with a biological question. You don't develop technology for the sake of technology. You develop the technology to be able to answer a question. But that technology then enables biology in a very much more fundamental way. And the prototype there was work that Lee had done in B-cell rearrangement. So as you probably know, Lee actually won a Lasker Award for working out how B-cells rearrange the B-cell receptor and how antibody diversity is generated. But he needed, I think he was sequencing Benz-Jones proteins in the beginning, and so you needed to essentially develop a sequencer to be able to sequence Benz-Jones proteins, and then after that he needed to essentially sequence the DNA, and it developed the DNA sequencer, etc. But once that DNA sequencer had been developed, it then enabled the entire Human Genome Project. So essentially that was a good insight for me, at least as a younger scientist at the time, as to how technology could enable biology as well. Rob asked me to discuss just one or two ways in which we've used technology and essentially the influence of Lee on how we came to apply that. And I think at the beginning we'd been talking about blood as a window on health. That's been Lee's mantra for as long as I've ever known him. And I had been working on macrophages, which are one of the white blood cells of the innate immune system. And they interact with every part of the body as they circulate through. And I thought, well, if we could figure out the barcode of information that exists within the macrophage, we'd be able to tell whether or not that went through when it went through the pancreas, if there was a little bit of cancer beginning to develop there, etc. We'd be able to extract that information as an early diagnostic. But as time went on, we began to develop essentially a network analysis of macrophages in the context of various infectious agents, in the context of a whole series of inflammatory diseases and so forth. And the two studies that I wanted to mention was, one, we wanted to try to understand how tuberculosis is reactivated. It's actually an amazing fact that TB, a third of the entire world is infected with TB. That's 2.3 billion people. It's latently infected, and when the immune systems drop, they become active. And the question is, how can you actually figure out, if they are going to become active and when they're going to become active? Because that's a really critically important insight to be able to have. The reason for that is that you can then isolate them. You can, before they go active, you can prophylactically treat them, and you can have much shorter therapeutic sort of times that you need to. At the moment, you need to treat people for 6 months before, and that essentially is involved in driving drug resistance. And now we had in TB, we had multi-drug resistance, then extreme drug resistance, and now total drug resistance in India and in Africa. So shortening the course of treatment is critically important. And essentially, I took just basically took all the technology Lee had developed, some of the insights he'd given me, went to South Africa, which I'm from originally, took 12,000 kids who were latently infected and followed them out for 12 years, taking blood every few weeks, and then looking at transcriptomes, because we knew what the macrophages transcriptomes looked like under various conditions. We actually managed to figure out a diagnostic for TB, which would predict TB going from latency to activity. And at that time, we'd also we established, if you remember, a systems nanotechnology consortium, again, one of Lee's ideas, which was Lee, Jim Heath, Steve Quake, Mike Phelps, Mike Rokas, and Charles Sawyers and I. And essentially developed some of the early technologies which would be able to be used in the field very cheaply to try to measure these signatures that would, in this case, predict whether people were going to go from latent to active TB. And at the same time, again, based on an idea of Lee's, went to South Africa to use cell phones. This is 15, 17 years ago now. And what we used them for was to essentially transmit digital information about X-rays to Johannesburg, which was the center. Many people can use a X-ray machine, but very few people are capable of actually interpreting the X-ray. So the nurses would take X-rays, send the information via cell phone to the center. They'd be read, and then they would the break would either be set or not. And so really, when I think back of this, and this is, Rob just told me a few minutes ago what he wanted me to talk on, and it was such an easy thing to just grab a whole series of things and say, this is what I got from Lee, and he's had immense influence on me. I've walked away now as head of an institute across the street which looks at infectious diseases. We're very interested in vaccine development, and I think rational vaccine development is within sight, all due to Lee's insights in technology, and I thank you for that.
Well, Rob also put me on the spot and asked me to talk about what I've learned about technology development in the time that I've been doing this, and in the context of introducing Lee. So I've been thinking about how to address this, and I decided to go back pre my career in technology development to what I learned in my PhD. And I was very fortunate to be able to interact a little with some just terrific scientists, some giants in the field, and so it seemed appropriate to talk about that in this context. And I knew what I learned scientifically in my PhD, but since I had a Doctor of Philosophy, I asked myself when I finished, well, what did I learn in terms of philosophy? And so after thinking about that a little bit, I decided I learned three important things in my time as a PhD student. And these came from three bits of advice from three different scientists. And the first was a piece of advice from Cesar Milstein, who was the head of my department at the time. And obviously he was instrumental in the field of developing technology for monoclonal antibodies. Anyway, he asked me about my PhD project and what I intended to do. And I think he wasn't very impressed, because he proceeded to give me the following advice. He said, what you should do is do something really that's at the limits of your ability. You really need to challenge yourself. Don't do something that's too easy, too straightforward, because if you do that, you can't really make a big impact on the field. So I took that to heart, and I've been suffering from that ever since. But when I met, I think in a fairly early conversation with Lee, he was telling me about his ambitions to launch this institute and his ambitions in this area of systems biology. And I thought, my goodness, if anyone is taking Cesar's advice, it's Lee. I mean, it's clearly, it's such a wonderfully ambitious goal. And it really, I think, it speaks volumes more than anyone can say if you think about, taking on a challenge like that. So that impressed me greatly. And I think an ability to do that really comes down to well, there are a lot of important things. But another thing that impressed me about Lee that I think was important is that Lee is very open-minded and very sort of Catholic in terms of his willingness to talk to anyone. I know that because at the time I was in between and trying to figure out what I was going to do, and he was willing to spend time to talk to me. And I think if you want to take on these grand challenges, you do need to have the really terrific things happen at the interfaces of different disciplines, and you have to be able to be very open-minded and work with very diverse groups of people and bring diverse groups of people together to solve those problems. And obviously Lee has done that. The second piece of philosophy I learned in my PhD was, at times things weren't working and it was very discouraging. And a piece of advice from Fred Sanger is that if it works at all, you're in good shape because you can make it work better. And I remember once at a museum I saw an exhibit of the very first Xerox, and it's just an awful looking thing. But Xerox now is a beautiful thing. So obviously, that's a great example of something like that. And I think, again, experts will often, because they know so much, be able to tell you why something won't work or why it's a bad idea. But often that means missing a startling new way of thinking about something. And so, I've always, I think, been encouraged in talking to Lee in that he's always thinking about how to solve problems, not how, why things won't work. It's important to be rational, but it's also important to, to be able to basically be positive and figure out new solutions to hard problems. And I don't know that systems biology, it seems to me it has a lot of potential in terms of coming up with new ways of thinking about how to do systems biology. It seems like we're still figuring that out, so it's very, very exciting from that point of view. And the third thing I learned was from Max Perutz. And he said one time, I remember him saying at a seminar, that it took him 30 years to finish his PhD project. And he was just grateful that they didn't require him to finish before giving him his PhD. And his point was that, if you're trying to tackle big, ambitious goals, you need patience. You can't expect necessarily to solve things overnight. And in his case, he changed the field of structural biology. So 30 years, in retrospect, I think most people would say is not a lot of time for the amount of impact that he had. It was just tremendous. And so, one thing I've admired about Lee is his determination. I think he's unstoppable. And I think that's one of the things you need to be able to tackle these grand challenges. So the final thing I'll say is that, in the technology field, you're always dealing with disruptions. I remember a great Dilbert cartoon that I saw somewhere, and it said something about technology. Technology is not for wimps. So disruptions in technology, I don't think we should be afraid of. Sometimes, they present new challenges, but it's great for the field, and it creates great opportunities for figuring out new ways to do things, and it's something we should take advantage of.
Thank you. So this is, somewhat of a learning lecture at the moment as well. So this is all about trying to learn something from Lee in this unique situation that we have here at the moment. And so Lee is going to get up now. He's not going to have any slides, and he's going to give us his perspective of emerging technology. So the one question I really had to start this off, and maybe you'll answer this throughout your talk, is really, when do we really understand disruptive technologies, and whether we should be developing or whether we should be embracing them, and whether this is something learned or is it innate? And for me, it's something more learned, and maybe Lee will give a better perspective on that. So, Lee.